Imagine looking at the Earth and trying to make sense of millions of different life forms all at once. Without a way to group them together, studying biology would be almost impossible. We need classification. That means arranging living organisms into specific groups based entirely on their similarities and differences. Humans have been trying to organize the natural world for thousands of years, but those earliest attempts were totally instinctive, driven by our basic daily needs for food, medicine, and shelter. If we look at this ancient cave painting of horned beasts, we see humanity's first rough drafts of classification. People simply divided nature into highly practical categories. Animals were either dangerous or useful, and plants were either edible, medicinal, or poisonous. Grouping a tiger as dangerous and a cow as useful definitely kept early humans alive. But this system had zero scientific basis, and it completely ignored how these organisms actually functioned on a biological level. This marble bust captures the Greek philosopher Aristotle, widely known as the father of biology. He was the earliest thinker to attempt a genuinely scientific method for organizing life. His method relied heavily on morphological characters. Morphology simply means looking at the external form and visible physical structure of an organism, its size, its shape, and its distinct body parts. Using this visible criteria, he divided all plants into 3 straightforward groups: large woody trees, medium bushy shrubs, and small soft-stemmed herbs. He applied a similarly stark logic to animals, splitting them into 2 groups: those that had red blood, which roughly maps to modern vertebrates, and those that did not, like insects and earthworms. But judging organisms purely by their appearance is a dangerous trap. If you rely entirely on morphology, you end up grouping whales alongside fish simply because they live in the water and swim, entirely missing the biological reality that whales breathe air and produce milk. To solve this confusion, Carolus Linnaeus, the father of taxonomy, developed the highly structured 2-kingdom classification system. He placed every known living thing into either kingdom Plantae or kingdom Animalia. For macroscopic life, this worked beautifully. It clearly separated trees, mosses, and ferns from insects, birds, and humans. But by zooming in past what the naked eye can see, microscopes revealed millions of tiny rule breakers that shattered those 2 neat categories. This diagram highlights the massive biological divide between 2 distinct cell types. On the left, prokaryotes, which are primitive cells completely lacking a true nucleus, and on the right, eukaryotes, which are advanced cells containing complex, membrane-bound organelles. Linnaeus's 2-kingdom system forced both of these entirely different structures together, arbitrarily grouping primitive bacteria into the plant kingdom alongside highly complex eukaryotic trees. A classification system built only for human eyesight completely collapses the moment you examine cellular architecture. This image shows an amoeba proteus. The 2-kingdom system grouped single-celled organisms like this into the same broad categories as massive multicellular animals. And look at this vintage illustration of fungi. Taxonomists originally threw mushrooms and yeast into the plant kingdom, ignoring how these organisms actually survive. Green plants are autotrophic. They manufacture their own food using sunlight. Fungi are entirely heterotrophic. They cannot make food, relying instead on absorbing nutrients from dead or living organic matter. Then there is Euglena, a microscopic organism that possesses plant chlorophyll, but moves and acts like an animal. With all these contradictions piling up, dividing the world into just plants and animals failed to explain biological diversity. Scientists needed to abandon superficial traits and find hidden structural criteria. In 1969, an American ecologist named RH Whittaker proposed the 5-kingdom classification. Whittaker threw out the old playbook. Instead of just looking at outside appearances, he based his system on rigorous scientific metrics: cell structure, body organization, reproduction methods, and mode of nutrition. This photograph captures a dense cluster of rod-shaped bacteria. Under Whittaker's system, bacteria were finally pulled out of the plant kingdom and given their own dedicated space, Kingdom Monera, the exclusive home for all unicellular prokaryotes. Next, he created Kingdom Protista to house the single-celled eukaryotes, like the amoeba, and he established Kingdom Fungi for heterotrophs that have distinct cell walls made of a tough substance called chitin. That left the original macroscopic groups clearly defined: Kingdom Plantae for multicellular organisms that make their own food, and Kingdom Animalia for multicellular organisms that consume others. Whittaker's system resolved the scientific crisis by organizing life based on internal biological mechanics rather than superficial appearances. If you ever need to remember the order of those 5 groups, just use this simple memory trick. My pretty friend plays athletics. That stands for Monera, Protista, Fungi, Plantae, and Animalia. But Whittaker also looked toward the concept of phylogeny. This is the idea that true classification must map the evolutionary history and shared ancestry of organisms over time. Because biological science never stops evolving, later scientists proposed a 3-domain system that further splits Monera into 2 distinct branches, archaea and bacteria. As we can see here, modern taxonomy is no longer a set of static filing cabinets. It is a dynamic, interconnected map showing exactly how every form of life on Earth is related to one another. Humanity's quest to classify life started out as a primitive, instinctual tool just to survive in the wild. Today, it has become our ultimate blueprint for understanding Earth's vast biological history.